Today's lecture is on vaccines and biotechnology. Edward Jenner was the first person to do a vaccination, and he actually realized that milkmaids back in 1796 seemed to be immune to cowpox. So he figured that there must be some type of protection that they were allowed due to being exposed to cowpox. So he did a little experiment where he actually took a boy. We don't know who the boy was, but basically he inoculated him with cowpox. Luckily the boy recovered. Then later on infected him with smallpox, and the child did not show any symptoms. So smallpox really isn't a disease that we see, and if you take a look at this little guy here, you can see what smallpox actually looks like. So the CDC estimates that from 1994 to 2013, in the US alone, vaccines prevented 21 million hospitalizations and 700,000 plus deaths. Again, there's a lot of controversy right now, as you guys know, about vaccines. Some people don't think you should have them. But things that popped up back in 1998 had to deal with a link that was said to be between the MMR vaccine and autism. So basically, guys, what happened after that, a lot of people decided not to get their children vaccinated. Turns out this study was false, guys. The authors were funded by lawyers of parents that had autistic kids. There were lawsuits that were filed. It was a major conflict. And actually, the main author lost his medical license amid the charges. So again, there is definitely no link between MMR and autism. In studies following that, guys, thousands of participants were involved, and in 2015, actually 95,000 children were shown to not have a link between MMR and autism. So again, if you take a look, guys, measles and pertussis have shown a very strong reemergence. In fact, in 2015, Disney had an outbreak, 189 cases, and also it turns out that this was over 24 different states. So again, by people not getting vaccinated, we're clearly seeing an increase in the number of these cases. So again, there's a lot of things that have occurred since that time. A lot of schools say that there's no enrollment, there's no more opt-out for personal reasons. California actually requires children to have at least a 10-day immunization to attend school or daycare, and only in cases of medical exemptions are these going to be allowed. Now again, you can take vaccines in various ways: injected, inhaled, and ingested. And basically they may consist of an attenuated microbe, which is weakened, parts of a microbe, isolated and inactivated toxins, and also genetically manufactured portions of a microbe. So they don't provide immediate protection, which a lot of people don't realize. It takes about 2 weeks for an antibody to be at its peak. So basically what happens is your vaccine is going to stimulate an immunological memory, and then when you become exposed to that, your memory cells act quickly to prevent the infection. The CDC provides all the vaccination recommendations. You guys know that from your discussion posts. Routine childhood vaccinations protect kids against more than 15 different pathogens. And you want to stimulate an optimal immunological memory, so a lot of times booster shots are also going to be needed. And they have to be spaced apart because your B cells and T cells need time to actually do their thing and provide you with your protection. So again, some parents prefer to space out vaccinations. Problem with this, guys, is it breaks down herd immunity. Herd immunity, guys, is when the majority of people are vaccinated, and that actually offers a protection for others. So if you do not get your kids vaccinated, they're not going to be fully protected. So for your exam, please know that there are different types of vaccines. One is called live attenuated, that will contain altered pathogens that do not cause disease but are still infectious. Benefits is they stimulate a potent immunological response. Drawbacks, they can cause disease in an immunocompromised host. Guys, we're kind of seeing that right now with corona, unfortunately, that the people most likely to get sick are going to be immunocompromised, elderly, kids, etc. Another one to know, guys, is an inactivated vaccine that consists of a whole inactivated pathogen. Whole agent vaccines, guys, will have the entire pathogen and it's inactivated by heat, chemicals, or radiation. Now, subunit vaccines, guys, they don't include the whole pathogen. One of the problems with some of these is they require what are called adjuvants, like ammonium salts and monophospholipid A. That's to make it big enough for your immune system to actually recognize and do its thing. The drawback with this is some people have allergies to those components. Okay, so we're going to move into now a little bit more diagnostic stuff. So for your test, know that serology is the diagnostic testing of serum. And a lot of the tests that we'll talk about rely on that. So one reaction that you can take a look at is called agglutination. And that's when antigens, antibodies come together in clump. So in regards to red blood cells, guys, we call that hemagglutination. Okay, that's agglutination of red blood cells. Please know that for your exam. And this just shows, this is what you'll see if something is clumped up, and down here, that's when it does not agglutinate. The next test I'd like to talk about, guys, is called an ELISA test. You have a virtual lab that you're responsible to do for this. And this stands for enzyme-linked immunosorbent assay. And key things to know, there's two types. One type will detect antigens, and we call that a direct ELISA. And the second is an indirect ELISA, and that will detect antibodies. So again, guys, your direct ELISA detects antigen, and indirect detects antibodies. And another really good test, guys, is called PCR, or polymerase chain reaction. And what this does is it amplifies the amount of DNA in a sample. And you can actually amplify up defective genes to look for genetic disorders, diagnose a lot of different types of infection this way. So one key thing I'd like you guys to know for your test is that we use what's known as Taq polymerase. Know that, Taq polymerase, heat stable. So we have to put that in there because when we cycle through, we have to heat our samples up very hot. And if you use regular polymerase, unfortunately, it just would not work. And here's the process, guys. You start with your DNA. You've got to heat it up to separate your strands here, guys, and then start to copy the new DNA. Then you have several different copies. You keep cycling this through, guys, usually up to about 35 cycles, which gives you 2 to the 35th power copies of DNA when you start with one copy of DNA. So for your exam, guys, I would like you to know these steps. We have a melting step, and basically at the melting step, guys, you raise the temperature to over 95 degrees C. That's to separate the double-stranded DNA. We also have an annealing step. In the annealing step, guys, you will lower your temperature, usually somewhere between 50 and 65. That will allow primers to bind. And the primers are important, guys, because in the extension step, which is the third step, you raise the temperature a little bit more, between 65 and 75, and that will allow then the Taq polymerase to bind to the primers, and that will copy the target DNA.